Hello, everybody. We're going to get started. Thank you so much for joining us today. Today we have on the line Dr. Bray Darpo, who is the Chief Scientific Officer at ERT. He's also worked in industry on the sponsor side. And very important for this discussion, Dr. Darpo led the IQ-CSRC study, which validated the concept of applying exposure response analysis on data from early stage clinical trials to replace the TQT study. So we're happy to be speaking with you today. We're going to move forward with the presentation. If at any point during the presentation you have a question, please feel free to type that up in the Q&A box. And at the end of the at the end of the presentation, we're going to give those questions over to Dr. Darpo. So thanks again, and we're going to get started. Go ahead, Dr. Darpo.
Thank you, Kelsey, and thanks to all attendees for sharing your valuable time. I'm happy to present today one of my favorite topics, how to capture ECGs as part of early clinical development, and therefore or thereby potentially waive the request for a therapeutic study. So I will share some background to this evolution. Since 2005, things have moved, and then since 2015, we are now allowed to apply concentration QTC analysis, which really what triggered our ability to look at early clinical studies as a replacement for the therapeutic study. So the regulatory scene, while I turn off my cell phone, the regulatory scene has changed, and this change was much based on two major components. First of all, regulators and also, of course, industry and sponsors have gained an enormous experience on applying concentration QTC analysis on their early studies and also on their therapeutic studies, and learned how concentration QTC results relate to the typical by time point analysis in the therapeutic study. And I'll give you one example of this. And then the second component was that the FDA asked industry and the CSRC consortium, which is the Cardiac Safety Research Consortium, to run a prospective study looking at a number of mildly QT prolonging drugs to determine whether a small size study could detect the effect of a mildly QT prolonging drug. So since December 2015, a lot of things have happened. Today, I would claim that most therapeutic studies are done based on concentration QTC analysis, and therefore with a much reduced sample size as compared to some years earlier. With the intersection union test, which is the statistical approach used for a by time point analysis, we were typically looking at 40 to 44, possibly more subjects per period or per group in a therapeutic study. Today, we're looking at 24 to 28, so roughly half the number of subjects. And secondly, in many first-in-human studies, serial ECG monitoring is paired with PK sampling, and then concentration QTC analysis is applied either on the SAD or the MAD, or actually on the pooled analysis. And potential effects of the drug are analyzed this way. And within certain pre-specified criteria, these data can serve as a replacement for a formal therapeutic study later on during clinical development. Also, an informed approach is to, since most sponsors they don't have an exact knowledge of the PK of their compound when they take it into first-in-human, and therefore it may be exactly known how much of accumulation you will see upon multiple dosing, as an example. So with unknown PK, it often makes sense to implement the procedures, collect the ECGs and store the waveforms, and then make the decision what and when to analyze once PK and other project considerations are better known. So this is one early example of concentration QTC analysis applied on a typical, very standard single dose study. We're looking at 6 dose groups with 6+2, so 36 on active and 12 on placebo. A linear model was applied, and here we are looking at the change from baseline QTc on the y-axis as a function of increasing plasma levels on the x-axis. And of course, as you can see by the spread of the data, there's much less data up here in the high range. But you can also see that the predicted effect, which is the black line with its confidence intervals in gray, the predicted effect is very shallow, and there's no indication that the effect will exceed 10 milliseconds, even up at very high concentrations. And these data come from the Salicystat development program, and the data were presented for EMA some years ago, and the sponsor was granted to continue their development program into late-stage clinical trials, pivotal trials, without performing a thorough QT study. Sometimes it is also a clear advantage for a sponsor to know whether their drug causes QTc prolongation early in development. So there may be indications based on non-clinical data, and that may justify performing this analysis as soon as you have the data at hand. The sponsor had seen that the drug may cause QT prolongation from other MAD studies, and therefore performed a tailored MAD study. And you can see here that the predicted effect, the black line, has a positive slope, and the confidence intervals are quite narrow with that positive slope. And the data, the red boxes are the data, are captured very nicely by the model. If we, as an example, assume that the clinical relevant plasma concentration is around 600 nanograms per ml, the drug will on average cause, in the population, on average cause, say, 16 to 80 milliseconds QTc prolongation, which would be a concern for regulators and for the risk-benefit assessment. So we did this IQ-CSRC study. It was actually planned 7 years ago in 2013. And the study was planned in close collaboration with industry. As you can see in the paper, there are several industry clinical pharmacologists on the paper. The CSRC, which also represents industry, but also academia and service providers, and FDA. So FDA gave us a long list of mildly QT prolonging drugs, and from that list we chose 5 drugs without much of heart rate effect, but with known QT effect. So we looked at ondansetron, dolasetron, quinine, a low dose dofetilide, and moxifloxacin. And we enrolled 20 male and female subjects in the study, and each subject underwent 3 treatment periods. We used an incomplete block design, meaning that 9 subjects in total received each active drug and 6 received placebo. So the data I'm about to show you compares 9 on active with 6 on placebo, so very small numbers. We looked at 5 positive drugs, the ones I just listed, and 1 negative drug, levetiracetam. Each treatment period consisted of 2 days, and on day 1, a dose intended to give around 10 to 12 milliseconds QTc effect was given, and on day 2 a somewhat higher dose. And the ECG methodology we used in this study was what is called today the expert precision QT technique, and the study was sponsored by iCardiac, which is now part of ERT. And of course, these data are published. And this is how moxifloxacin looks when we're looking at the effect as a function of increasing plasma levels. You see a clear-cut positive slope. The fact that the slope is positive can be seen by the confidence interval here. The lower bound is above 0. And then we can look at the observed mean Cmax of the 400, the lower moxifloxacin dose, 400 milligram. And we see that it's close to 1.9 microgram per ml. And at this level, the predicted effect is clearly above 10 milliseconds. And then we're looking at the upper bound. So if we look at 1.9 nanograms per ml, we can see that the confidence interval here is clearly above 10 milliseconds. So moxifloxacin met the criteria we had established to confirm that a small study could detect a mild QTc prolongation. Ondansetron was a bit more challenging, which can be seen by a more shallow slope, still still it's a positive slope and somewhat wider confidence intervals. And when we look at the Cmax after the low dose, 284 nanograms per mil, we can see that the gray shaded area is clearly above 10 milliseconds. So ondansetron, being the second positive example, also met the pre-specified criteria for detection of mild QTc prolongation. Levocetirizine had undergone 2 previous therapeutic studies and have been shown to be negative. And this is how levocetirizine looks with 9 on active and 6 on placebo. A very shallow black line, a shallow slope with quite wide confidence interval around the estimate. And you can see that throughout the concentration here on the x-axis, the gray shaded area is nowhere near the 10 millisecond threshold. So thereby we have demonstrated that an effect exceeding 10 milliseconds can be excluded within the observed range of plasma levels, so up to 1200 nanograms per mil. We can certainly say that this drug does not cause any clinically relevant QTc prolongation. And this is how it looked. All 5 drugs came out positive, and levocetirizine, as I show here, came out negative. So these data were available in August 2014, and 1 month later, we shared the data with FDA, but also with the ICH E14 working group. And at a public meeting in December 2014, the E14 group decided to start looking into whether it was feasible to allow concentration QTc analysis to replace the by time point analysis. And this is how it looks. This is how it looked when this revision, which is a revised Q&A section of the E14 document, came into place in December 1 year later, so December 2015. And the first point I've highlighted here is that if sufficiently high plasma levels are achieved by the drug, then a separate positive control would not be needed. And this is critical because in our first in human studies, we will not have a positive control. But on the other hand, in the experience of many sponsors, in the SAD study, the SAD part, we often achieve very high plasma levels as compared to clinically relevant. And therefore, this is a good opportunity to demonstrate that the drug does not have an effect. Secondly, the same criteria as we used in the IQC-SIC study was taken into the guidance. So if you can show the upper bound of the two-sided confidence interval is below 10 milliseconds at clinically relevant concentrations, then this is the same as to say that you have a negative QT assessment. And if you then have achieved sufficiently high plasma levels, then these data can replace the therapeutic study. So how much does the ECG methodology as such matter? Does it matter if we use a semi-automated approach, which most core labs would, a manual approach, which still can be seen sometimes, or the expert precision QT technique as the technique that we use? Does it really matter? And of course, when you have a drug like this, and this is actually not a drug, it's a food ingredient, a sweetener, monotin. If you have an effect like this, you don't need much of precision. This sweetener causes clear-cut QTc prolongation. You can see that the predicted effect is up in the range of 20 to 30 milliseconds with very narrow confidence intervals. And if you have a drug that causes a pronounced QTc prolongation like this, precision doesn't matter really much because you will see the effect even with a cardiologist and a ruler and a pen. But what we would like to do is to exclude that our drug has an effect in our early studies, and then precision matters, because the better precision you have, the more narrow confidence intervals you will see around the predicted effect, the black line. And the more narrow this gray shaded area is, the more likely you are to exclude a 10 milliseconds effect. And this is again levocetirizine. So then precision matters. And that's why I'll show you some main differences between a standard semi-automated method, which we also have, and we use it mainly in patient studies, and the EPQT ECG methodology. So with a standard semi-automated method, most core labs today use a Holter, a continuous recording. And from this continuous recording at pre-specified time points, the reader will extract 3 strips of ECGs, so 3 10-second strips of ECGs, sometimes reported by software and sometimes just visually. From these 3 strips, 3 ECG beats are chosen. And then each of these beats, or in total 9 beats, are looked at by an underlying computerized algorithm and then adjusted by the technician. So we end up having measured 9 beats with a process that is highly overseen by the technician and or by the cardiologist. This is a very robust methodology. It's well fit for patient studies where we more often have T-wave morphology changes, and it's a widely used and well-recognized method that's been in place for more than 20 years. But we also know that it comes with a certain level of variability, of course. The EPQT process also uses continuous recordings, and we extract up to 10 replicate ECGs using an underlying computerized algorithm, which we call TQT+. Then the underlying computerized algorithm called iCompass categorizes each beat from these 10 strips. So up to 100, 120 beats are categorized into high confidence or not so high confidence. And the high confidence beats, and those are classified this way because of discernible end of the T wave, relatively stable heart rate, good signal to noise ratio, etc. All the high confidence beats are accepted, whereas the not so high confidence beats are looked at by the technician and either accepted or rejected. So there's never any adjustment of measurement points or fiducial points. So all accepted beats goes into the equation, and we end up having measured around 90 to 120 QT-RR beats or heartbeats, measuring the QT and the RR interval in up to 100 beats per time point. And this then translates into a lower variability. This is a study we did in collaboration with Novartis and Karin Meiser, one of their lead statisticians, some years ago. And it compares the variability, that is the variance in the model, compares the semi-automated method in red with a expert precision QT technique, at that time high precision QT technique. And whether you compare drug versus placebo or moxifloxacin versus placebo in a crossover design or in a parallel design, you can see that the variance with the expert precision with the EPQT technique is roughly half of the variance when you use a semi-automated approach. These 7 studies were conducted at the same site with in principle the same protocol and certainly with the same study procedures. So what we're comparing here are 7 different studies with 2 different techniques. And these data clearly show that the expert precision QT technique reduces the variability quite dramatically. And this can also always be paired up with greater numbers. So if you are to conduct a therapeutic study in concentration QTc, it may well be that you can do the study with a semi-automated approach on 36 to 40 subjects instead of 28. But when you take this into first-in-human studies, we don't play around with the numbers that way. We take what we have, so to say. And if we assume that the drug has a small underlying effect, and the typical assumption is that the drug causes a 3 milliseconds average effect, and if we then use a EPQT technique with a standard deviation of the change from baseline QTc, so a variability of 6 milliseconds, then we need 9 subjects on active and 6 on placebo, which is also demonstrated by the IQCSRC study. Whereas if we use a good semi-automated method, we come at a standard deviation of change from baseline between 9 and 10 milliseconds. And if it's 10 milliseconds, it would require roughly twice as many subjects, so 21 subjects on active and 6 on placebo. So precision has a clear role when it comes to excluding a small effect. And these are the numbers you need to have in order to demonstrate that this drug does not cause a QTc prolongation exceeding 10 milliseconds using concentration QTc analysis. And these are also published concentrations and data. This is lemborexant, one early example of a program that was cleared without a therapeutic study and also approved without a therapeutic study based on concentration QTc analysis applied on early studies. And this is one of the MAD study, and you can see that this is looking at all different dose groups, and you can see that if you just focus on the upper bound of the confidence intervals, of course these upper bounds are sometimes about 10 milliseconds because we're looking at small cohorts. We're looking at 6 on active and 2 on placebo. But when we take these data, and actually these graphs show data from 2 first-in-human studies. From 2 MAD studies. One was the first-in-human MAD study, and the other one was the standard bridging study into a Japanese population. And when we pool these 2 studies, the scatter plot looks like this, so not so much data in the upper range of the concentrations, and this is how the model, the concentration QTc model, comes out. The slope is very shallow. The confidence intervals are wider, of course, at high concentrations because we have less data there, but even so, it's clearly below 10 milliseconds. This led the FDA to approve this drug with the following label in December 2019. And it states that in the concentration QTc analysis using data from 2 randomized, double-blind, placebo-controlled multiple MAD studies in healthy subjects, lemborexant does not prolong the QTc interval to any clinically relevant extent at 5 times the maximum recommended dose. And that is, of course, important. Clinically relevant concentrations are down to the 40 to 60 nanogram per mil level, whereas achieved concentrations was 5 to 6 times higher. So clearly, these 2 studies demonstrated that lemborexant at very high concentrations did not cause QTc prolongation, and therefore the data were accepted without a positive control, and that also went into the label. The lemborexant development program was cleared without a therapeutic study, not only in the United States, but also in Japan some months later and in Europe a few weeks later. So where should we place our ECG assessment in order to potentially replace the therapeutic study? And this is all about pharmacokinetics. You should place the evaluation in the part of the study, in the SAD or the MAD, depending on the PK of parent and metabolites. Often sponsors push the dose more in SAD studies using 6, 7, 8 dose groups, whereas they then may only take 3 doses into the MAD. In cases, in the majority, not vast majority, but majority of cases, you will see higher concentrations of both parent and metabolites in the single ascending dose study part. But sometimes the drug has a long half-life, or metabolites have long half-lives. Look at some of the tuberculosis drugs as an example. And then you need to look at the effect in the MAD study, because only there you will see sufficiently high plasma levels of the metabolites or of the parent. So this comes down to question in regard to PK. And as I've already said, since PK is unknown at the start, often unknown at the start of the first-in-human study, it makes sense to implement the procedures, that is to collect the waveforms and make sure the subjects are supinely resting at pre-specified time points, and then store the data and to decide which part to analyze, or possibly both parts, depending on the PK and also depending on the project viability, of course. So what will it take to obtain a therapeutic study in summary? First of all, you need to be at least 2-fold above Cmax in patients, mean Cmax in patients, not looking at individual outliers, but looking at mean Cmax in patients with impaired clearance of the drug. That means, as an example, if you have a 3A4 substrate and you have a twofold increase in mean Cmax in patients on itraconazole, as an example, a potent 3A4 inhibitor, then you need to be twofold above this mean Cmax. So, which often then would mean fourfold above the average patient. So you need to target and you need to focus on patients with impaired clearance, either based on intrinsic factors such as hepatic or renal impairment, or extrinsic factors, that is, drug interactions. And when you cannot achieve a sufficiently high margin, that calls for a study with a positive control, which in my mind pushes the evaluation into a surrogate study. Now, there is an ongoing discussion within the ICH group on additional non-clinical assays and standardization of non-clinical assays, and that may, at least long-term, have an effect on which fold of concentrations you need to demonstrate that a drug does not cause a relevant QTc prolongation. Sample size is not really a subject for regulatory decisions. It is the problem that has to be handled by the sponsor. If you have too small sample size, you will not be able to exclude an effect. Your confidence intervals would be too wide. One thing that I think is worth pointing out is that often, well, not often, but sometimes you end up having only 4 or 5 or 6 subjects on placebo in the pooled placebo group, and that makes the study or the evaluation very vulnerable for random findings. So typically, and also regulators are aware of this, typically to protect against false negatives and false positives, we typically recommend having at least 8 subjects in the pooled placebo group. So is there then a remaining role of the surrogate study? And I think there are 2 main scenarios. The first one, of course, that some sponsors choose to conduct a surrogate study only for drugs that make it beyond proof of concept or early phase 2. But the results have to be available before you initiate your phase 3 study. And then secondly, when sufficient exposure margin cannot be achieved or was not achieved in the first in human study or any other study in healthy subjects, then you need to do a study. And often that would be a study with positive control, which in my mind is a therapeutic study. Sometimes also, but perhaps more of an exception, the sponsor doesn't really find the data from the first in human study credible enough to believe in it, or they find a small effect which they would like to either refute or confirm, and then they perform a surrogate study. And with this in mind, we still conduct surrogate studies. And I'll come back to one example. Another way to improve the confidence in the data is applying what we call the method bias sensitivity test. This was a test that we developed in collaboration with FDA, as can be seen from the publication. And without going into the details, what we're doing here is that we're comparing a fully automated ECG methodology with what the core lab does. And if we in the process see that we introduce bias, a negative bias, meaning that prolonged QTc intervals are measured short, then there would be a slope in this Van Alpen graph, and that slope needs to be small enough to demonstrate that you can have confidence in the data. This is what we call the MBS test, which we often apply in our first in human studies. And since we are using so much data from ICOMPAS, the underlying algorithm, we very rarely see that the bias is large enough to be worrisome. We have seen it in a few patient studies, and that makes sense because in patient studies you need more of an overview by a cardiologist who more often than will change what the underlying algorithm does. So that makes sense. But in healthy subjects, we rarely see that we introduce a bias with EPQT. So an update on the ICH discussion, and I covered part of this. I think the largest part will be a discussion on the S7B and how to standardize non-clinical, both in vitro and in vivo assays, but also how to perform risk assessment based on non-clinical assays. It may be but this is an ongoing discussion that much of this is already covered by at least large sponsors, and it may be that for large sponsors this may boil down to a change in the standard by which they perform the Hurg assay. Maybe that a dynamic Hurg assay makes sense to perform. What is the impact on E14? Well, I think it's one really good item is that oncology will be discussed in more detail in the future Q&A, which I think we will see a public version of for comments at least within a few months. There will also be a discussion on a sufficiently high plasma concentration range when you're doing a study without a positive control, which I think is a good step forward. And overall, the text that relates to E14 will promote the use of first-in-human studies to replace a therapeutic study. Then the FDA, as you probably know, also advocates the use of exploratory ECG biomarkers such as JT-peak assessment for cardiac risk assessment. I think it's fair to say that this JT-peak will not make its way into E14, and other regulators remain quite hesitant as to whether this biomarker is useful or not for risk stratification, or perhaps I should phrase it this way, whether we have experience enough to take this novel biomarker into a regulatory guidance paper. QT assessment oncology is often performed without placebo and can be done in a phase 1 study in patients, often with solid tumors, either in the dose escalation phase or in the extension phase. This is one example from the extension phase with a dose that the sponsor took into patients. So once the MTD dose was defined, it was taken into the extension phase, and this is a ECG assessment performed on that extension phase. It's an open label study in 18 patients, and you can see a somewhat positive slope, but you can also see that an effect exceeding 20 milliseconds, which is the threshold for oncology, can be excluded. This means that without placebo and with a 20 milliseconds threshold and with concentrations that are only clinically relevant because this is the clinical dose, the sponsor will be able to claim that this drug will not cause a more pronounced disease prolongation, so we can exclude a 20 milliseconds effect, which is very often good enough in oncology because the indication is life-threatening or vital, and therefore the small QTc effect is not seen as the same level of concern as in other indications. So my conclusions are that early QT assessment is now an accepted way to confidently exclude that a drug has a clinically relevant effect on the QTc interval. It has been the case that we can use early studies to say that, okay, this drug causes QTc prolongation. There's no need to perform a therapeutic study. You just need to find out what you see in patients. But now we are actually allowed to exclude the small effect and therefore replace the therapeutic study. I don't think the therapeutic study will completely disappear as a viable option in select cases, and I've covered those cases where we see sponsors still performing therapeutic studies. The revised E14 will further promote the use of early clinical studies, but also non-clinical data to characterize the pro-arrhythmic effect of a drug. And overall, I think QT assessment and ECG assessment has been made much more effective by the development we've seen over the last 5 years. We can perform therapeutic studies with much smaller sample sizes, and we can even replace and make the therapeutic study redundant by applying this technique into early phase clinical studies, which are planned anyway. So this is a part of a standard plan first-in-human study. And thereby, thank you so much, and I'll be happy to take questions.
Sure, Dr. Daza, we have a couple that have come in. One is, can you please elaborate on how to determine a sufficiently high multiple of the clinically relevant exposure?
Yeah, I'd be glad to, because that is actually the most common reason for sponsors and regulators to discuss. it's almost always discussion because the sponsor wants to claim that what they've seen is good enough and FDA pushes back. But what you need to do is to look, you to know, there's a document called the Highlights in Clinical Pharmacology document, which is a template document that can be downloaded from RT's website. In that document, it's detailed what you need to look at. And the emphasis there is to define which patient group will have the highest concentrations. And I gave you one example. If you have a 3A4 substrate, you will need to look at the mean Cmax in patients with a standard dose, but also on a potent 3A4 inhibitor. If you have a 2D6 substrate, you look at poor metabolizers to conclude what the mean Cmax will be in poor metabolizers. Then when you have defined this, you have defined the worst case scenario in terms of mean Cmax in patients, then you need to be clearly above this threshold. And formally or publicly, the statement is that you need to be twofold above this mean Cmax, but as you can understand, this is often a discussion, and you can push the envelope and claim that if you're above the mean Cmax representing worst case scenario in patients, that should be good enough. And also bear in mind, if we discuss mean Cmax in the first in-human study, the range of plasma concentrations will of course be higher than the mean Cmax, so there's room for discussion. But the principle is that you need to be above the worst case scenario in terms of mean Cmax in patients with impaired clearance. I hope that clarified the question.
Thank you, Dr. Darpo. And the next question is, I would appreciate your insights on preclinical data that would trigger concentration QTc in a SAD study. For example, what QTc prolongation in a dog CV study?
I see the non-clinical package as signal generating. If you have an in vivo dog study that shows, or a non-human primate study that shows QTc prolongation, I think it emphasizes that the sponsor needs to look at this in your early studies. So in those scenarios, I think it's quite mandatory to perform careful ECG assessment in your early study, not only using safety ECGs, but actually look at potential effect as a function of concentrations. So in my mind, it emphasizes the importance of applying concentration QTc analysis in early studies when you have a non-clinical signal. If you don't have a signal at all, you can make the informed choice whether you want to apply it or whether you want to do a therapy study. For a small sponsor, it's often a clear advantage to know that their candidate drug, does not have this liability to present when they enter negotiations with potential partners. For large sponsors with many compounds in the same indication, it may also be useful to know which one has no QTc effect. So there are a lot of different considerations that go into this, and the non-clinical part is one way of getting more information, knowing more how to strategize the ECG assessment, and make decisions as to whether apply EPQT or any concentration QTc analysis into early studies.
Thank you. The next question we have is, are there other therapeutic areas besides oncology where more liberal QTc threshold is warranted?
I'm sorry, Kelsey, where the threshold, I didn't...
Apologies. So is there...
Oh, I see. Okay, I see the question. Thank you. A more liberal QTc threshold, like 20 milliseconds. Well, in principle, you can argue that this would be the case in any life-threatening condition. I've only seen the 20 milliseconds being applied for drugs intended for cancer indications. I haven't seen it, as far as I recall, in any other therapeutic area than oncology. And I think it would be more challenging. But in principle, this would apply to any life-threatening condition where there are no good alternatives.
Thank you. The next question is, do regulatory agencies require method bias sensitivity analysis results as part of a waiver application?
No regulator would request it, but FDA performs MBS as a test within their warehouse, so they do it routinely. And of course, as a sponsor, I think it's better to know what they will see than being told what they will see. So I would typically advocate applying MBS, method bias sensitivity test. It's a minor effort to do. It's a minor cost that adds to the study, and it gives you greater confidence in the data. But it is not a regulatory request. But as I said, it is routinely applied by FDA as shared by Lars Johannesson some years ago on a public meeting.
Thank you. Next question. How many waivers have been granted by the FDA, EMA, and PMDA? I don't know if you know that off the top of your head.
Well, I don't know it off the top of my head, and I know Dr. Kleiman tries to monitor this, try to look at approved drugs. So we know there are several examples, but I can't really give you a number. Dr. Garnett from FDA gave an update on this, but this was 2.5 years ago. And by that time, they had approved 11 projects without a therapeutic study outside oncology then. But that was 2.5 years ago. She also said that they're seeing an increasing amount of studies or examples where concentration QTc analysis is applied to early studies. But those were the numbers 2.5 years ago. So the only thing I can say is that it's clearly more today, but I can't give you an exact or even an inexact number as how it looks today.
Great. And the next question is a full time-matched baseline day required?
There's only one example where a day minus one, if we by that the drug-free baseline day is required, and that is when the drug has a substantial heart rate effect. So if the sponsor learns from the non-clinical study or perhaps by the therapeutic class that they can expect a substantial heart rate effect, and that is typically defined as a placebo-corrected change from baseline of more than 10 beats per minute. So either in the bradycardic or more often tachycardic direction. Then you need to calculate an individualized QTc correction method, QTc. And for that purpose, you need a 24 or 12-hour baseline. So in that example, you need day minus one. In all other examples, so in a single ascending dose study or a multiple ascending dose study, you do not need the day minus one. You do not need a time-matched baseline. One exception, further, in a parallel designed therapeutic study, you always have a baseline day as the baseline, and then you for both placebo and active drug. But since we're doing less and less of these these parallel design therapeutic studies, that is something we see less often today. But that's also an example where you need a day minus one. So the drug has a substantial heart rate effect. Yes, you need it. If you're running a parallel design therapeutic study, yes, you need a day minus one. In all other examples, in the SAD, in the MAD, and the crossover design, it's good enough to have a pre-dose baseline derived from 3 separate time points.
Thank you. Next question is, how long do subjects need to be supinely resting at each nominal extraction time point?
But we never studied this in a systematic way. So we have a standard language, and that standard language is that subjects need to be supinely resting for 10 minutes prior to the nominal time point and 5 minutes after. And that is just to make sure that we have a window during which the subject is supinely resting for at least 5 minutes before we start to extract ECGs. I think most core labs would use a similar language, but we never studied this systematically. What we see is that in the few examples where we have tried to extract ECGs without the subjects being strictly controlled, then we see much poorer quality. We see much larger variability, and sometimes we're not even able to extract the number of replicates that we're looking for. And then we need to do a semi-automated approach for that time point, and even so, we see much more variability. So subjects should be supinely resting, and we advocate 10 minutes prior to dosing, and 10 minutes prior to the nominal time point, and 5 minutes after. Sometimes in more complicated first-in-human studies, there are so many study procedures that you have interference between this supinely resting time point and other study procedures, and then we can reduce the resting period to some extent, or try to place other study procedures at other time points, so to say. But it's ideal to have the subject supinely resting at least 5 minutes before the extraction starts. So that would be 10 minutes in total before the nominal time point.
Okay, and the last question I have here is, how much exposure will enable us to avoid separate positive control as per ICH E5? I'll send you this question. Is this only applied to a drug with clinically relevant effect on the QT interval at sufficient high exposure?
Can you scroll up so I can see the start of the question, Kelsey?
Sure.
Why I'm looking at this is because the ICH E5 is something else. I would assume that there should be ICH E14 Q&A question 5, but I'm not sure. If it's the latter E14, then I think I've covered that. I think it's twofold above worst case scenario. One question that often comes up in regard to other ICH guidances is the fact that in Europe, most IRBs and regulators are more hesitant to allow doses to be pushed too high in the first-in-human study. But in our experience, if you justify and if you perform your single ascending dose study, and if you justify the need to assess ECG effects at high concentrations, in most cases you will be successful in not only getting approval for your protocol, but also completing the study. So we haven't really seen that as a major problem, even though some of these studies, of course, are performed in Europe. We haven't seen that this surfaces as a problem very often.
Great. And I think we only have time for a couple more.
Perhaps I should also add, this is always a question of tolerability. No one and no regulator would ever expect sponsors to intoxicate their subjects. So if you have a drug that is poorly tolerated, this is a justifiable reason. This is a good reason not to push the dose, but also the inability to perform ECG assessment at very high concentration. It may mean that you pushed into study with a positive control, that is a therapeutic study, which today can be done with a relatively small sample size of 24 to 28. But no regulator would ever expect us to intoxicate our healthy subjects. So intolerability is a good reason to argue in front of regulators. This is as good as it gets.
Great. Thank you, Dr. Darko. And I appreciate all the questions. They're coming in very, very quickly. There's a lot to get through. So I will say, if we don't get to any today, your question today, we can certainly follow up after, but we'll try to get through a couple more of these. The next one, Dr. Darko, is the DDQTC ECG using individual corrected QT K-1 ECG profile, principally superior to baseline adjusted CDQTC. So we butchering that?
Well, I'll try to interpret the question. So we're comparing QTCI with a standard population-based approach, such as QTCF, and I think it's been very well clarified today that if the drug doesn't have much of a heart rate effect, no one expects you to do anything but QTCF. So QTC corrected by Fridericia, which is a population-based correction method using a slope of 0.33. For drugs that have a substantial heart rate effect, you are expected to perform some sort of QTCI. Historically, QTCI was often derived from supine resting time points in most cases, or in many cases. And we know today that this gives you a QTCI that is pretty much like QTCF, so it doesn't buy you anything to do that. So that's also why QTCF, Fridericia correction, in most cases are fully accepted. Does QTCI reduce the variability? No, not in our mind, not in our experience, not if you're looking at drugs without much of a heart effect. A more relevant and a question that is more often discussed is whether you should perform hysteresis adjustment for drugs with a heart rate effect. Hysteresis adjustment would take into account the heart rate during the preceding 1, 2, or 3 minutes, and then correct for that heart rate rather than the immediate heart rate before the QT interval. And I think that's still up in a good topic for discussion, and still something that we are actively looking into to see how much that buys us and how much that reduces the variability. So I think there are cases and there are places where we can still improve our ECG assessment, and perhaps adjustment for QT or hysteresis is one such example for selected cases. And the cases I will go for first then are, of course, drugs with a heart rate effect. But besides this, and all standard cases where the drug does not have a heart rate effect, QTCF is as good as anything else. What you never should do today is to calculate QTCB, because we know for sure that is a biased QT correction method. So Bassett's correction should not be used.
Thank you. And we had another question on if you could expand on why the method works, why we would do this in healthy volunteers, this method specifically as compared to patients. And then interpreting that as APQT specifically in healthy volunteers.
Yeah. Well, in my mind, if you can do an ECG assessment in healthy subjects, you should always do it, because it's first of all, I mean, these studies to achieve the level of precision you need to do this study with a patient, with a subject in-house. It has to be in-clinic, in-house study. Secondly, the underlying notion here, which is widely agreed upon, is that if you don't see a QTC effect in healthy subjects at sufficiently high plasma concentrations, you will not see a QTC effect in patients. There is no good examples of patients as a group having QT prolongation when you have not been able to demonstrate this in healthy subjects exposed to sufficiently high doses / concentrations of the drug. And that's why we do it in healthy subjects, because it's a great opportunity and much less costly to do it in healthy subjects as compared to patients. It's much less costly and much more efficient. And also you have great precision. The variability in patients will be higher. And of course, patients with cardiac disease will have the highest variability, but also in general, patients, including cancer patients, have higher variability. So the small numbers we're looking at here cannot be repeated in patients for sure. Am I convincing?
I think so.
Thank you, Kelsey.
We have a question on the selection of high and low confidence beats, and does that introduce bias?
I think that's a very fair. I think that's a fair question. Yeah, I think that's a very fair question. Do we introduce bias by doing this? Well, that's why MBS, because MBS will look at the full background beats and compare that with the core lab output. So if we introduce bias, we will see that by performing the method bias sensitivity. That's why we do it. And to date, and we have done this for 3 years now, to date we haven't seen one single example in healthy subjects where this has been the case, whereas we have seen at least one and possibly two patient studies where the core lab procedure, the EPQT, actually introduced bias because there was so much of T-wave morphology changes that everything had to be had to be repeating using a semi-automated approach. But those cases are patient studies where you have more often have T-wave morphology changes, making the measurements more difficult.
Great, and I think that we have time for one more question. For studies and compounds that have not been granted a waiver, what are some reasons?
I would say, if I were to respond very quickly, not having achieved sufficiently high plasma concentrations. And that is still also, and that was also the case when we did traditional surrogate studies. The most common debate and argument between regulators and sponsors have always been whether you have achieved sufficiently high plasma levels and or doses when we did look at plasma levels. So, and that is the most common reason. And that was also quoted by Dr. Garnett at the meeting 2.5 years ago, that the most common reason for disagreement between regulators and sponsors were the achieved plasma concentration levels.
Wonderful. Thank you so much. So, folks, we're at the top of the hour here. We will have a recording available of this session. If you would like any additional information, feel free to reach out. And the questions that have not been answered, we will have somebody reach out to you directly or get you more information. We really appreciate the participation and hope you learned something with us today. So thank you for joining and have a great rest of your week.
Thank you so much.